Hello guys, good morning to all. Today I am here to give a lecture on canal system of Porifera. To know about Porifera, first we have to know about the animal kingdom. This animal kingdom is divided into vertebrate and invertebrate. These invertebrates is then divided into 9 phyla. What are these? First one is protozoa, then Porifera, then Coelenterata, then Platyhelminthes, then Escalminthes, then Annelida, then Mollusca, then Echinodermata. One, two, three, four, five, six, 7, 8, 9. Arthropod. So of these 9 phylum, Protozoa, Porifera, Coelenterata, all of these are particularly live in the watery, that is they are aquatic in nature, aquatic in nature, they are aquatic in nature, and most of them are particularly present in the marine or fresh water. So Porifera, why they are called Porifera? Because they in their body they have pores, so they are called pore bearing animals, which was first discovered and coined by a scientist known as Robert Grant in 1825. Their body is two-layered, and that's why they are called diploblastic. Outer layer is called pinacoderm and inner layer is called choanoderm. Beside this, as they have the pores here, these pores are called ostia, and if the large pores is there, this is called osculum. This is the picture of the bentham zone of a sea. Bentham zone means the lowermost zone where have the substratum. So as they are sessile, because they have no locomotory organ, they can attach with the substratum like the seabed, they can grow like this. So outer pore is called ostia and this pore is called osculum. Beside this there is a large gap. Besides this there is a large gap which is called spongocele. And outer, this is called pinacoderm, and this is called choanoderm. These choanoderm layer have some flagellated cell. These are the flagella. These are the flagellated cell. And as these cells are present here, this portion is called flagellated chamber. And all this structure, this structure is called spongocoel. So, outer structure have the pores. So, water from the sea moves through here, then enters here, then passes out through there. This is called the canal system. So, canal system means these are the components of the canal system. What does it? First one is ostia, then incurrent canals, then radial canals, then prosopile, spongocoel, apopyle, osculum, choanocyte, and pinacocyte. See this picture. This is spongocoel, and this is radial canal, and this is incurrent canal. So radial canal and incurrent canal is jointed by prosopile. Again, this radial canal join with the spongocoel by apopyles. And through this water is moved inside the spongocoel and this move out towards. So as the osculum, choanocyte, prosopile. So prosopile is the opening between the incurrent and radial canal, and this from radial canal to spongocoel is called apopyle. What is incurrent canal? What is radial canal? These are the canal which move outwardly. This is called radial canal, and which move inwardly. This is called internal canal. Now, there are three types of canal system. Now, one is Ascon type, then Sycon type, then Leucon type, and then Ragon type. First one is Ascon type. See here, Ascon type is, this is the outer layer, pinacoderm. This is the innermost layer, choanoderm, having flagellated chamber. This is the outer layer having the pores called ostia. Ostia, again, these ostia means pores. These again present in a cell called porocyte. The cell containing a canal-like structure is called porocyte, and this is called ostia. So water moves through this ostia, passes through here, and ultimately reaches into the spongocoel, and due to the action of flagellated chambers, flagella present in there, water is moved from the outside to the inner side to the spongocoel, and again due to the action of this flagella of the flagellated chamber, they are pushed outside. So Ascon type, it is the simplest type, and main example is Leucocoelonia. I have already described it. The wall is perforated by, it is called ostia. Now, next one is called Sycon type. So, as evolution is going on, so due to the evolution, the structure of the particular sponge from Leucocoelonia become most complicated. At the same time, the structure of the, suppose this is the Ascon type. So after evolution, this structure is very much folded and ultimately form a structure called Sycon type. So this is the Sycon type, where you see this is the radial canal, this is the incurrent canal and spongocoel. So this is the from incurrent water from outside enters into the incurrent canal. then into the radial canal through prosopiles. Then from prosopiles, the water moves to the radial canal. Radial canal is supported by choanocyte layer, which containing flagella, and as they are known as also flagellated chambers. These flagella also moves the inward movement of water into the ultimately spongocoel through apopiles, and ultimately from apopiles it moves outwards through osculum. So here dermal pores, then incurrent canal, then prosopiles, then radial canal, then internal ostia, spongocoel, osculum, and then moves outside. So this is about the cyconate type of canal system. These are the thing: ostia, already told you, ostia, incurrent canal, then prosopile, then radial canal. And while radial canal is also called flagellated chamber, because radial canal is filled up with flagella cell, that's why it is called flagellated chambers. And this is apopyle, then excurrent canal. One thing, radial canal then open to the excurrent canal, then excurrent canal then moves to the spongocoel. So between the spongocoel and radial canal, there is another canal known as excurrent canal. So the movement of water from outside into the body or spongocoel of the particular sponge is called incurrent water, and the water which moves out from the spongocoel to the outside through osculum is called excurrent water. Through this water, all the physiological activities taken out, which I will describe later on. Now this is the third type of canal system, which is called leuconate canal system. So leuconate canal system, as this is the again more and more complication occurs. That means this can be broken down into, this flagellated chamber is broken down into small oval rounded like structure, and ultimately forms a peculiar type of canal system called leucon type of canal system. How it forms? Due to the breakdown of incurrent canal and radial canals, and ultimately radial canals form small lobules, and which is bounded by flagella, and that is, these are called also flagellated chamber, these radial chamber. And at the same time, as it is diploblastic, this is mesenchyme or mesoglea is there, this is also broken down, they are deposited here, and ultimately form a particular type of canal system, which is called leuconate type of canal system. So in leuconate type of canal system, similarly the water moves from outside, this is called dermal pores, then to the incurrent canals. Many incurrent canals and many radial canals are here in case of leuconate canals, because they are totally broken down to form many incurrent and radial canals. So they move from incurrent canals, they move through prosopile or prosodas into flagellated chamber, that's means radial canal, then radial canals through apopiles, they move to excurrent canals, and from excurrent canals by internal ostia, they move here, spongocoel, the main cavity or gastrocavity, from where it moves outside. Next one is, last one is called, this is the leucon type. So already told you, the further folding of choanocyte layer, radial canal, evaginous to form many chambers, oval shaped chamber, and mesenchyme, which is completely break down to make the space between them. So both incurrent canals and prosopile canal, they are become rounded shape in prosopiles and flagellated chambers. Why they are called flagellated chambers? Due to the presence of flagella, and all this from. So this is all about. So there are three types of canals, three types of leuconate type of canal system. Due to the presence or shape of flagellated chambers, In case of Urophilus, there are three types: Urophilus, apodal, and diplodal. In case of Urophilus type, the flagellated chamber is thimble shaped. An example is Tatilia. In case of apodal type, flagellated chamber is small and rounded. It is called stellata. And in diplodal type, the flagellar chamber are small and rounded, which is found only in Spongilla. Now the last one is called ragone type. Here it is completely separated from others. This type of canal system found in the only larval forms of sponges. Here, this basal portion is called hydrosphere and upper portion is called spongosphere. These hyposphere or hyposphere portion have no flagella or flagellated area, but these spongosphere have the flagellated area. And here subdermal space, these are called subdermal space, and from subdermal space then incurrent canals, then through prosopyle it moves to the radial canals. These are the radial canals, that means flagellated chambers are radial canals, they form radial canals. It moves through apopyles to excurrent canals, and then from excurrent canals it moves through internal ostia, through spongiosin, and ultimately moves outside. So, told already, basal wall is called hyposphere, and which is devoid of flagellated chambers, and upper wall is called spongosphere, and ostia open into the subdermal space is already shown. This subdermal space formed by breaking down of renal canal. The flagellated chamber lead to the spongosil and open by single osculum. Now, all these canal system, there are 4 types of canal system found in sponges. So all this, why this type of canal system occurring here? Because in case of us, we need circulation, respiration, excretion, for all physiological activities should be there. So in case of, as they are very small animals, so various physiological processes occurring due to the presence of canal system are respiration, nutrition, then circulation, and at the same time excretion and reproduction. So due to the water current moves through the body, there is exchange of oxygen and carbon dioxide occurring there. So this is about respiration. Second, in case of nutrition, all the materials with nutritive material passing through the canals and through spongosil, it moves out through the osculum. At the same time, the chambers or cell that take up the nutritive materials. At the same time, number 3.1 is excretion. Excretory materials can passes out through this flowing water, ingression and excretion of water through the osculum, excretion can be done. And last one is reproductive. The male gamete, which can be passes through the ostia and then through spongosil and can be deposited in the female gamete, and for successful external reproduction it can be done. So these are all about the physiological or function of the canal system. So this is all about the canal system of Porifera. In my next video, I will give a lecture on spicules of sponges. If you like it, you just subscribe it. Thank you.